about to start, and then the whole seminar will be recorded. So if more people want to view your talk, because I do hear lots of people are interested, they will have the access through the YouTube video that we will release by the end of today. So this time, welcome everyone to join the Wednesday seminar this week. And this week we have Luoth Chou, currently a postdoc at NASA Goddard Space Flight Center. And prior to that, she was a postdoc at Georgetown University. I first met Chou. I first met Lu actually was because I want to co-convene a session for AppSciCom, and then I approached to Lu, and she's super nice. And later on, she became a co-organizer with me for many activities, and we realized we have so similar research interests. So I'm very excited for this talk. And Lu, welcome.
Thank you so much. Okay, let's get started. I'll turn on my camera for easiness. So hi everyone. Thank you all so much for being here today, and thank you, Grace, for inviting me to speak with all of you. My name is Luoth Chou. My pronouns are she and her, and I'm currently a postdoc fellow at NASA Goddard Space Flight Center and Georgetown University. Actually, if none of the pandemic situation was going on, I would have actually been super happy to have been back on campus, on the UMD campus, because it's actually my alma mater. I graduated in 2013 with a degree across the road in a different department in microbiology and astronomy, and went on to do a PhD at U of Illinois at Chicago. So it's great to be back in the area, and hopefully for the long term, and it's just so great to be speaking with all of you today. And my talk will be on agnostic biosignatures detection using mass spectrometry. And I'll start by pointing out this, or doing this cheesy thing by pointing out this cosmic perspective that we all share, especially when thinking about astrobiology. And it's really because often the search for life elsewhere begins at home. And we all know this image of Earth, this pale blue dot that never fails to remind us that all of the life forms that has ever existed that we know of are sitting on this pale blue world. And it's also important to remember and acknowledge that this is truly the only example of life that we have. It's really a double-edged sword in a way, because on one hand, it's self-evident that life exists in the universe. We know this, we are the manifestation of that life form. But on the other hand, we have to place ourselves outside of this perspective and contend with a much deeper question. What is life elsewhere? What could it look like? Especially if it did not evolve or share the same heritage as Earth. So as astrobiologists, we have to be sure not to have what my colleagues like to say, these horse blinders on when we study life and when we design missions to look for them on other planets. We also have to grapple with our limited knowledge about the universal definition of life itself. So starting with this helps, of course, but one can't really define life without any dissenting viewpoints. And as Dave Desmarais, a prominent NASA astrobiologist, puts it really elegantly, if you have 100 people in the room, you're going to have 150 definitions of life. But even without a universal definition of life or an agreed upon operational definition of life, we can still use our observations and our intuition about what kinds of imprints that life leaves behind in the environment, and to begin to ask how we can detect life elsewhere. These imprints, called biosignatures, can come in the form of chemical signatures or physical signatures. Now, some of these chemical ones can be specific diagnostic compounds, or DNA or RNA that are great biosignatures, or biogenic gases like oxygen, or some of the more controversial ones like the methane on Mars, or the recent putative detection of phosphine in the atmosphere of Venus. Physical biosignatures, on the other hand, include things like the morphology of a cell, the spectral features of vegetation or pigments, which can be detected on remote exoplanets, or dynamic movements of particles that is inconsistent with non-biological process, or literally fossils. So let's go through some of the examples of what I call familiar biosignatures. So DNA, RNA, and proteins, we all know are central to life on Earth. They're used to carry instructions of the cell and provide an explosion of functionality on a biochemical level. So for example, protein of one sequence can fold a certain way to carry out a function in a cell. And just by changing one amino acid, it can fold a totally different way. And these are great biosignatures because the likelihood that things like DNA, RNA, or proteins form long strands by just pure abiotic processes without life is actually really low. But the building block themselves, the amino acids and the nucleic acids, for example, are quite common in the universe. And so there is always a caveat to consider when looking for these kinds of biosignatures. Now, another really great example is homochirality, which is the idea that all life uses one form of amino acid. Now, amino acids are chiral molecules, meaning that they contain in their structure a configuration, the shape of how the atoms are put together, essentially a mirror image. Like if you put your hands together, they are a mirror image of each other. They're not superimposable. And amino acids in biology only exists in one-handedness, whereas outside of biology, the ratio between the left-handedness and the right-handedness is 50-50. Now, this has been such an enigmatic problem in our understanding of the origin of life, and the question as to why life chose or accidentally ended up with one of these handedness is still an open question. Now, the same can also be said for isotopes, though we have a better idea of why biological processes are known to fractionate and produce distinct isotopic signatures. And by that, I mean biology likes to use molecules that are typically lighter. And as we know it, isotopes are elements of the same numbers of protons, but different numbers of neutrons, meaning that while there's the same chemistry, they differ by weight. And so we can use these kinds of isotopic biosignatures to infer the presence of life. Now, I call out these examples, though knowing that there's, again, caveats in each of their successes as biosignatures. For example, the plot on the right here, you can see my laser pointer, right? Okay, I saw a nod.
Yes.
So for example, the plot on the right here shows that in meteorites, there are some higher percentage of one-handed configuration, the one that's actually used in biology over the other. Does this mean that there's life in meteorites? Not likely, but it shows that some mechanisms could give rise to these kinds of biosignatures that we see of today. Now, another example that I thought might be familiar to some of you are morphologies. And so these things are called stromatolites. This picture here. So stromatolites are sedimentary structures or rocks formed from layers of photosynthetic bacteria or other microbes and sedimentary particles that have lithified over time. These stromatolites can express strange features that's recognizable in textures, and it's these textures that we use to identify life through simple morphological observations or petrography. But of course, like other biosignatures, it's faced with the uncertainties, especially when comparing it with other fabrics of rocks in the absence of biology. To a trained geologist, perhaps this looks like a stromatolite, but maybe it could possibly be, I don't know, a metamorphic rock after all. It's important to note, however, that all of these biosignatures that I've talked about so far, the familiar examples, are fundamentally tied to the history of life on Earth. What do I mean by this? So I'm showing here, I'm sure a lot of you are familiar with this schematic already. I'm showing here a geologic timescale that accompanies the evolution of life. Now, if you were to imagine being dropped off at a different time on this timescale, what biosignature would you detect? For example, oxygen didn't show up at considerable concentration in our atmosphere until about 2.7 billion years ago. So life had about 1 billion years to evolve extremely complex mechanism to produce oxygen. It would have had to evolve photosystems that are able to take energy from light and turn into oxygen at enough concentration that's detectable. And even then, oxygen shares this deep history with the evolution of life on Earth. And because of this, we posit that oxygen is quote unquote too specific of a biosignature. And as you can imagine, forms of life out there that produce something else besides oxygen. So let's go back to the drawing board and consider what are some of the biosignatures that we can use that are not tied to a specific biochemistry or that are historically contingent for a particular planet like ours. If we imagine biosignatures of life on Earth as this oval shape, we know that all of Terran biology express these biosignatures. However, we also know that some of these biosignatures are very specific to life on Earth, or life as we know it, but some may be more general. Now, these more general biosignatures could potentially be features of life that are consistent across the universe. And so we call these universal biosignatures or agnostic biosignatures. Now, as you move away from this universal biosignature circle, the biosignature becomes specific to that planetary body. And so if you have other planetary bodies that have other history and other ancestry, other origin, or simply different geochemical evolution, we could in theory have exotic biosignatures that are specific to that body. And we know that each different planetary bodies experience a different and independent geochemical evolution, for example, Mars, and likely driving their biosignatures to look differently than Terran-based biosignatures, unless, of course, we share that same ancestry. But again, some of these features of life, the life that we are, could potentially be more common. Therefore, if we focus our attention on the center of the circle, or at least drive our search strategy towards that center, we have a higher chance of capturing unfamiliar life, which importantly also includes Terran-based or Earth-based biosignatures. So in summary, universal or agnostic biosignatures are signs of life that do not presuppose an underlying biochemical system and is agnostic to the planetary condition that we're exploring. Now, I want to dive in a little bit further onto what we can do as a community to enhance our search strategy. We know that within this dichotometry, yep, is that how you say it? Yeah. Between Terran-only biosignatures and universal biosignatures contains our knowledge, or what we know in this yellow oval, of what are the expressions of life that we can recognize, some of which are likely universal, where it overlaps with this universal biosignature circle here. And some of this knowledge is captured by our ability to detect those expressions, this blue oval here. Now, the area that overlaps between the yellow and the blue shows detectable recognized Terran biosignatures, what we know of traditionally as the biosignatures that we care about, that are specific chirality or biosignature gases or molecular fossils, for example. And now we propose that our search strategies or efforts should focus on where our detection capabilities overlap with the knowledge on universal biosignature. And we can do this by broadening our knowledge base, which essentially boils down to increasing the size of those two ovals and focusing our efforts on where it overlaps with the darker gray circle. Now, this can be done via two different approaches: the top-down approach, where we would examine the existing data and determine key chemical or physical differences between life and non-life, and then test these findings against a reference sample set that span the chemical space between biotic and abiotic. And the other approach is the bottom-up approach, which essentially encompasses research and development, including modeling, theoretical, and experimental studies on universal principles of life, biophysical laws, complexity, as well as the origin of life. Now, these studies would provide us with a deeper understanding of what are the possible chemical, physical space that life occupies and how we can use that knowledge to enhance our detection capabilities. And very importantly, these two approaches would be used to corroborate and iterate the knowledge so that we have a much greater understanding of how to detect potentially universal features of life. Okay. Now, we also know that to enhance our detection strategies, we also need to expand our detection capabilities. Now, I highlight the first step of doing this in this talk, which is by taking advantage of existing high heritage instrumentation to perform broader science measurements. These other recommendations can be found in the white paper that my colleagues and I wrote to address the other strategies that we can implement towards a more universal or agnostic biosignature detection strategy submitted to the 2022 Planetary Decadal Survey. So email me if you'd like a copy of this paper. And so let's dive into it. So I'll focus specifically on the detection capabilities in terms of flight heritage, where low heritage is an instrument that has not yet been developed for flight, but is being considered. And high heritage are instruments that have been miniaturized and optimized and have actually flown on previous missions. And so one of those things are mass spectrometers. Now, mass spectrometers is a great example of high heritage instruments. They've been used in space exploration since the 70s. They've been miniaturized, optimized, and flown to help us understand some of the most important discoveries in planetary science and potentially the search for life elsewhere in the universe. They're also critical payload in future missions like ExoMars and Europa Clipper and JUICE, and of course my personal favorite, Dragonfly, which is set to explore Titan. Now, Dr. Arevalo here at UMD has a very strong program and involvement with NASA missions concerning advancing mass spectrometer technology, so I certainly encourage you to talk with him to learn more. And so we know that traditionally mass spectrometers are used to detect biosignatures such as complex molecules that could be present on the surface. For example, these molecular fossils here. Now, these compounds, like these fatty acids or the sterines here, used in organic geochemistry typically for paleo-environmental reconstruction, for example, are quite robust Terran biosignatures because their presence and distribution require a suite of complex biosynthetic pathways to produce and to produce in high abundance. So detecting one of these specific patterns or this molecule here is almost an unequivocal sign that we detected life. There's just no question about it. But this contemporary interpretation is very specific to Terran biochemistry. And so we propose that there are alternative ways to interpret the data which we can get back from mass spectrometers, whether the ones that are on board current missions or future missions. And one way to do this would be to instead look at their intrinsic complexity, where molecules like short-chain fatty acids here may have a much lower complexity or information, and molecules like this sterine here has a high complexity, meaning that the probability of it existing randomly is very low. So this would suggest the presence of life or lifelike machinery that are able to catalyze these reactions, forming these compounds, but without the knowledge of what that particular biochemical system is. And we can do this, we can approximate the intrinsic complexity by using its mass spectrum. Now, I don't want to talk about this work because it's still currently in development by the University of Glasgow in partnership with us at NASA Goddard, but I'll go over some of the other methods that we're considering. So while we know that mass specs, like the sample analysis at Mars, which is currently on board the Curiosity rover, have successfully flown and been implemented, we argue that it's important to consider alternative strategies to collect and interpret the data, all of which would inform the development of future mass spectrometers. The sample analysis at Mars, or SAN, is capable of performing pyrolysis and gas chromatography mass spectrometry, and is able to identify molecules up to 500 Daltons, some of which could be those complex molecules that I mentioned. On the other hand, the Mars Organic Molecular Analyzer, or MoMA, is able to do what SAN does, as well as laser desorption and tandem mass spectrometry at a much higher mass range. Now, this capability allows for the fragmentation of higher molecular weight molecules, providing mass spectrum for which we can explore chemical complexity to understand whether some molecules, however complex they are, require the presence of life or lifelike machinery to produce. It's also important to note that the similar prototype of the MoMA instrument will also fly on board the Dragonfly mission, and that instrument is called the Dragonfly Mass Spectrometer. Yay! Another method that we can leverage mass spectrometry for, and which I'll focus on specifically today and show you some results for, is polymer detection using mass spectrometry. So, I think I alluded to this earlier, but we know that polymers play an important role in life on Earth by allowing it to access a larger chemical and functional space. For example, proteins help catalyze many more critical reactions than, say, an abiotic system. Polymers also allow for the reliable storage and propagation of genetic information. And so we think that polymers play an important role in life elsewhere, but we don't want to presuppose their underlying biochemistry, essentially presupposing their chemical makeup, whether they're based on nucleic acids or amino acids. But because we only have the examples of polymers on Earth, or the biopolymers on Earth, we have to start building our methods around them, and then try to agnostify our strategy while loosely defining the criteria of what makes up a monomer. Furthermore, while we would love to just look for polymers in space, we know that abiotic polymers, or polymers that are not produced by life, are actually quite common in the solar system, especially if you look at places like Titan and beyond, probably. And so we propose to develop tools that can detect polymers by identifying their monomer composition, like their numbers and compound class or functional groups, for example, and then use tools like machine learning to classify them potentially as abiotic or biotic. Although I won't touch on that last part today, since it is a huge feat to design an entirely new life detection system. And so I'll just go over what we have done so far in this particular project and walk you through some of our polymer detection method. So the first step that we need to do is generate the data, especially if we're going to use machine learning. Now, it's quite easy to do this with a benchtop instrument that mimic our flight instruments, but there's only so much data that we can collect from relevant samples. So we thought that we could start with simpler spectra by essentially artificially generating them. So we wrote an algorithm that generates the fragmentation masses of polymers in a mass spectrometer using a defined monomer set. And a good place to start, as I already mentioned, was amino acids, since we're very familiar with how peptides fragment in a mass spec, but this algorithm can be applied to any other arbitrary monomer set. We also collected some experimental data on a commercial laser desorption ionization instrument that is an analog to MoMA that will be on board ExoMars rover set to fly, I believe, next year. The next step would then to detect if there are polymers in the sample and potentially what are the monomer composition. We know that if polymers break at every monomer pair at covalent bonds, it would produce a signal that contains harmonics represented by the masses of those monomers. And we think that this information can be extracted using signal processing tools like Fourier transform or autocorrelation. And I'll only show you some results for autocorrelation today. So here are some of the results for that work. Maybe I'll try to slow down a little bit. So I'll first show you the artificial data for two sequences of the peptides making up of these amino acids here, the AK sequence, and then this sequence here. And you can see the masses of each of the letter in the key on the left here. These plots on the left show the artificial mass spectra with mass to charge on the x-axis and the relative intensity on the y-axis. The plot on the right are its autocorrelation function plot with the lagged step on the x-axis and the correlation values on the y-axis. Now, autocorrelation essentially compares the spectra to a version of itself by moving through these lag steps to see if there are any repeating patterns in the data. So you can already see from the ACF plots that all of the monomers in this artificial spectra, the A and the K, can be detected. And we were pretty successful at identifying monomer composition using autocorrelation made up to 5 letters within the sample, some of which could potentially even be dimers. We then applied this method on experimental data that we collected from that benchtop MoMA-like instrument. These are biological peptides, so not very agnostic. But in some cases, like for this peptide called bradykinin here, it doesn't work that well. We were only able to observe 2 of the monomers, but we were quite lucky with angiotensine, which is a peptide that's used to actually calibrate our instruments. And we were able to observe almost half of the monomers present using just this method alone. So those are the cases for known biopolymers, but what about other kinds of synthetic polymers, right? I mean, peptides we all know and love because we study them routinely in proteomics, for example. But what about other synthetic polymers? So here you can see that for this synthetic peptide, which is actually, yeah, it's a peptide, but I don't think it's a biological peptide. We can detect all of the monomers and even some dimers and possibly functional groups. And for something common like a paraformaldehyde here, we were able to detect formaldehyde monomers, dimers, trimers, and tetramers of this particular polymer. All of this is to show that autocorrelation is relatively successful at detecting monomers with the mass spec of a polymer sample, and that this process can be relatively automated. But as you can see, there's still a lot of work to do. And I'll go over some of that work, future work soon. So while we work through our proposed detection method using signal processing, like this autocorrelation function that I mentioned, making strides towards agnostifying it, we also want to benchmark it against machine learning. Now, machine learning should be relatively common at this point, and I know Grace is a huge proponent of machine learning, so I'm excited for her to be listening to this, to some of this development that we're doing today. So we developed a machine learning algorithm that allows us to make predictions about the identity or masses of the monomers in a sample. And of course, we focus on the masses because we'll be using mass spectrometry data. And so to do this, we use an algorithm called the multi-layer perceptrons, where we train a suite of classifiers, or each of these algorithms, to predict a monomer of interest in the sample. We then attempt to recover the accuracy of the monomer set size and the length of the polymer, and a couple of other factors that relates to what that polymer is and what it's made out of. Again, our starting point is using amino acids for now, which we know is not agnostic. It's based on Terran life. But we just simply want to see how easy is it for ML to help us make these predictions, and how easy is it to break these algorithms on just purely artificial spectra. We also are exploring different ways to introduce complexity to the data, for example, adding artificial noise and potentially denoising the spectra to develop some of the things that could happen in an actual mass spectrometer. And so we train this machine learning algorithm by training a suite of classifiers, as I mentioned, one of each for the monomer of interest. We subdivide the training set by 80% and the testing set by 20%. And then we ask the question, is this monomer present or absent in the spectra? So it's a binary prediction. And we then cross-validate the test data to get around 89% accuracy. So this is still in the works, but I just wanted to show some cool results and nice plots. Our pipeline then can be used for classifying entirely unknown spectra. And so this pipeline here, which is the classifier library that contains all of the trained classifiers, introduce an entirely unknown spectra, and then get the predictions afterwards. Great. So while we have some pretty good preliminary results, we know that we still have a lot of work to do. And so for future directions, we hope to develop an accuracy metric for signal processing strategy. So that's the autocorrelation work. And because right now we're able to observe known monomers, we know what we're looking for, but we don't have a specific way of defining its accuracy just yet. And we also have to determine the best way to agnostify it. What happens when we don't know what we're looking for? Of course, this is the crux of our research, but that's something that we're considering for future directions. We also want to start generating new sets of data for applying our machine learning algorithms to non-canonical polymers. So our classifier right now is trained on the 20 canonical amino acids, but we're planning on implementing a similar library of classifiers for masses that are within the range of our detection instrument. So basically one classifier for one mass. We also want to use artificial data built on arbitrary masses or masses of monomers that we expect in certain environments, like basically the building blocks of materials that we can observe in places like Titan, for example. And most importantly, we want to be sure that our method is robust against non-polymers. Now, we have not determined yet what are the false positive rates for our detection method, and so we're still exploring this super critical step in our analysis. Okay, I'm going really fast. I think I might finish this talk pretty soon. Sorry about that. Great, so to wrap up, I wanted to highlight that some of the methods that we're developing do work quite well for simple polymers, especially using autocorrelation, with some chemistry being observable, such as functional groups. We hope to test this on non-peptide polymers someday, and then on environmental samples, as well as lab simulated samples. We also noted that machine learning can be a really powerful tool when combined with mass spectrometry. I don't think I need to convince this particular group here, but we hope that in the future, we're able to implement machine learning for even further work, potentially detecting agnostic biosignatures in a sample or classifying biotic versus abiotic samples. And lastly, our goal is to verify these methods on flight instrumentation, especially the MoMA instrument suite on board the ExoMars mission and DRAMS on board the Dragonfly mission. And with that, I'd like to thank you all so much for attending the talk today. I also have to acknowledge some really incredibly talented co-authors and collaborators of this work. I thank my funding sources, NASA, a group at Georgetown, the Laboratory for Agnostic Biosignatures, and USRA for funding my postdoc. And I'm happy to take questions. I hope it wasn't too short. I was told I have an hour.
Thank you so much. I don't know if anyone else got excited, but I'm super excited. Is there any questions from the audience?
We can get personal. It's okay.
Okay.
Any questions, please, since it's a smaller group.
No, I have lots of questions.
Go for it, Chris.
I really, really like your talk, and our research are actually really, really matching to each other. So my first question actually comes from the drawing board that you show about the Terran biosignature, universal biosignature, and then why the importance of why we should aim for universal biosignature in order to search life beyond Earth. And I was wondering, what if there's no overlap between life on other planets versus life on Earth? Because you don't know what's the definition of life. In order for them to overlap, you are assuming to some degree that life elsewhere shares some characteristics with the life on Earth. But what if there's no overlap at all? And then the definition of life actually extends way beyond what we can visualize at this moment on Earth. Not even mention the evolution of Earth's history over billions of years.
Yeah, that is a really, really good point. And I think some of that is highlighted in that white paper that I spoke about. And you're absolutely right. I think as a community, we can acknowledge that as we go to search for life elsewhere, we have these horse blinders on. And so we want to take off those horse blinders in a way to be more inclusive to other potential life forms. But you're right. The question of whether or not a particular biosignature is agnostic, and there's a scale of agnosticism, right? Like it's agnostic, like very agnostic, where it's actually universal, versus agnostic as in, let's take a step back and consider maybe life uses polymers, for example. But life doesn't have to use polymers. We don't know that. That's still an open question. But it is one step removed from actually looking for the polymers that we know have evolved on Earth and are tied to our ancestry and biochemistry. And so, yeah, to me, I think that's assumption that we kind of have to make. And the fact that some universal biosignatures that we propose are still in question, because it's still an open question. It's still an open question whether or not we have any universal biosignatures at all, or universal traits of life at all, because of this lack of understanding or lack of a universal definition of life. I guess we could say it as humans because we're thinking about these things, and we, again, like, it's self-evident that life exists, right? But what is that life, and how do we begin to take off this horse blinders, like you're saying, and make sure that our search strategies is covering as much chemical space as possible. And so, yeah, I think it's still an open question. It's also an open question whether or not like life elsewhere would not share the same biochemical ancestry as Earth. It could very well be the same thing because there's something special about amino acids or something special about nucleic acids. And so those two different possibilities certainly exist. And I don't know if there's any way that we can answer those questions without studying the universal theories of life or theories of life in general and try to make it more universal. But of course, yeah, I don't think there's a way to confirm that until we find it.
So from your answer, you mentioned theory of life, but what is theory of life though?
Yeah, so theories of life, for me, is the branch of science that focuses on the biophysical laws or theories behind why life performs a certain mechanism. And so some of those things include evolution, growth and replication, metabolism is another one. And so all of those things are canonical features of life that have very deep theoretical reasons behind it. And so, yeah, I guess by theories, I mean, we think that life works a certain way. We develop these theories to explain that particular mechanism. This has nothing to do with biosignatures yet. It's just thinking about the mechanism behind life, whether it's dynamic, whether it has a certain feature that you can put on a plot and show that, or write an equation for, essentially. And so those theories of life, or the studies of the universal laws behind life, like why life behaves a certain way or why life shows a certain distribution, can inform us on how to find some of those features if they are expressed in a way that we can detect them, so chemically or physically, for example.
Cool.
Yeah.
So I got another question. So this method of measuring biosignatures with mass spectrometry and then using machine learning to try and interpret those spectra, that's sort of the way that we're trending. What other methods of quantifying peptides or biosignatures do we have in terrestrial lab settings? And is mass spec the best way to go? Is there some other DNA genetic sequencing that's probably better suited for this, or is mass spec just more easy and broad? Or
yes, great question. Yeah, so the reason why mass spec is the focus of our work is mass spec has a high heritage in space exploration. So we already have instruments that are built that can fly. That's essentially the main reason behind it. And of course, mass spec is used to sequence biopolymers on Earth. By biopolymers, I mean proteins, not nucleic acids and DNA, because we know that there are other sequencing machines that can do this. I actually have extra slides to answer to your questions. So, so another potential agnostic biosignature that we know of is, of course, DNA sequences. And so originally, the technology that we have come up with, for example, in DNA sequencing, have kind of advanced to these low power, low mass nucleic acid sequencers like nanopores. So they're just like basically a thumb drive that can sequence the DNA for you. And so traditionally, we would want to look for these long chain biopolymers or DNA or RNA and other samples. Again, if we find something like that elsewhere, if we find something like that elsewhere, we have to first ask whether that came from Earth or not. But if we find that elsewhere and reject that hypothesis, then maybe we know that we have detected life elsewhere that shares the same ancestry as Earth, that has DNA and RNA. Another way of using this particular technology that can do DNA or RNA sequencing is to look at surface complexity. And so by that, I mean, let's see if my next slide shows. Oh, it does not show. And by that, I mean, so if you focus on these two specific examples, we know that the surface of a cell has a lot of things protruding out of it. Like the cells express surface complexity, and it's these complexity that allows cells to interact with each other. Like we know that all cells on Earth have this particular biosignature, whereas minerals, on the other hand, have more simple surfaces. And so what we can do is actually design a method where short chain random sequences of DNA is bounded, or we add it into the solution that we think we're trying to look for, and then see what kinds of binding does it happen. If the surface material is highly complex, then we can make the assumption that there's potentially something complex that are in the sample. But if it's something like a mineral where it's less complex, then maybe it's not, maybe it doesn't have anything to do with life. And so this is another alternative way of using known technology, using something that we are really familiar with, and instead of looking for a defined biosignature that's restricted to our biochemistry, we're actually taking a step back and ask, what else can we use this for? And so, yeah, I hope that answers your question.
Yeah. So I guess an ideal mission, package would be if you have a mass spec to go in and look for some initial signs of life, initial organic molecules, and then maybe try and interpret that through AI. And then if you start to figure out that maybe you do have longer chains or longer peptides, then you can put it into this second one and dig deeper into what the sequence is. So that's kind of, okay, cool.
Yep. Yeah, essentially. And one of the other things too that I wanted to mention, that's kind of like the caveat of our studies, is that we could build ML or these principle first approaches using autocorrelation to detect polymers, but polymer itself doesn't tell you whether it's life or not. I mean, I have already mentioned that polymers exist elsewhere. But one way to actually tell the difference between a potentially abiotic polymer versus biotic polymer is through natural language processing. It's essentially looking at the information that's within those polymers and then see if the sequences of that particular polymer tells you anything interesting about where that's coming from, whether it contains information, if it's long chain enough, for example. But yeah, this is work that's being done by other groups.
That would be very cool.
Yeah, so I have a follow-up question to that. Can you go back to the slides where you show the autocorrelation and the machine, and then your example? Yeah, like here. Can you go back? Yeah, here. So how is autocorrelation different from Fourier transform? And then I actually still need some help to understand the figures on the left, on the right. How can you tell whether is A go prior to K versus K and A? How do you know the sequence?
So we don't, yeah. That's the thing about these methods, whether it's autocorrelation or Fourier transform, which are both kind of designed to do the same thing, which is to try to find what are some of the repeating features that are in that signal. So we use things like autocorrelation or Fourier transform to study like the fluctuation of CO2 in the atmosphere. And we can see that there's like an annual peak or whatever for, or a diurnal peak for day-night cycles. A very similar context here. It shows that the peak for A and the peak for K has a very high correlation with each other, because the difference between a peak that has that in it versus a peak that doesn't have it in it, where it fragments at that particular monomer, exists multiple times in the sample. But we don't know what the sequence is. There's other sequencing methods that are out there that other people are designing, but our focus is not on that just yet. That's a feat that's gonna have to happen a long time in the future.
So basically, the method design here is trying to understand the quantity of specific monomers that exist in your biopolymers. And then, is that correct?
I wouldn't go as far to say quantity yet. I want to be careful, or at least for me, because I don't want to oversell this. It's more of the prediction of what are some of the more highly correlated masses that show up in the spectra. And so really it's just presence or absence at this point. The fact that A and K happens to be equal to each other doesn't, I mean, it obviously we know what this is made out of because we made it, and we can see that A and K is equal to each other, but I don't want to, we haven't looked into whether or not this shows any quantification of the numbers of monomers in that particular sequence yet. So yeah, presence or absence at this point.
Okay, so when you load your sample into the machine learning network, so you have the classifier as your output, and then I don't really understand what is the input. Are you inputting your mass spectrum as a time series signal and then trying to understand better a specific class of molecule present from that specific time step, or you just input them as image and then try to classify them?
Great question. It's the first one. So we input them as spectra. So it's just an XY like plot, basically a 2D or 1D, 2D arrays. We have to bin them. That's the other thing about machine learning. In order for us to input into any particular classifier or perceptrons like this one, the input layers have to be the same size. And so we ended up binning them into 400 bins. This was originally done by one of our other colleagues. but we've also looked into binning them at 1200 bins, which is the numbers of masses that are in the mass spectrometer. And so those bins are input into these perceptrons, and then we also label the data. So this is supervised learning. We tell it that this particular mass spectra with this data, like with the XY data that I mentioned, has these amino acids or monomers that are in it. Can you tell me whether or not, and then we train that, and then you ask the perceptron, can you tell me whether or not there's A in this? And it says yes or no, and whether or not it has K in it, or G, or L, or whatever.
So when you train your layers, I assume each neuron in your input layer, there will be a synthetic data set that is only contain a single biomolecule or single class of biomolecule that you're interested in. And then you have the output layer that can classify yes or no. Is that correct? Because I'm having a difficult issue understanding whether the entire input layers is a single spectrum or actually a single neuron of the input layer is a single spectrum.
Yeah, good question. All of the data is inputted into the neuron. And so we generate the data for all of the experimental parameters that we want to study. So we have homopolymers, which are polymers that are made up of one type of amino acid. And then we select from a distribution of the amino acids and then generate that as a spectra. And so now instead of like a 2D array, it's just like a row in a CSV file, if we want to break it down to that. And then we would do this for, I believe, 50,000 spectra, and then use the binned masses as the input for the perceptrons. And so it learns for each of the row that we have these masses, we have these particular amino acids, and then this perceptron, the one specific perceptron for that particular monomer is going to learn from that, oh yes, I know that with these masses, I have this one that I'm looking for or not. And then we build the library of classifiers that contains the at least for amino acids, the 20 perceptrons that can make the predictions as yes, no.
I see, thank you.
Mm-hmm.
Okay, I have more questions.
Please.
What are the popular universal biosignatures that people are trying to detect right now, besides the complexity of biopolymers, the distribution pattern of monomers? I'm just wondering what are some of those biosignatures that we can explore with mass spectrometry or even beyond the mass spectrometry?
Yeah, so our team, the Laboratory for Agnostic Biosignatures based at Georgetown, is focused on 4 main themes. And so the first one is complexity, which is what I'm a part of. The second one is this, the surface complexity that I was telling you about earlier, which is called molecular complementarity. That's essentially looking for surfaces, the complexity of surfaces. The other one that I think are based on mass spectrometry is this thing called elemental disproportionation. And so it's the idea that cells tend to take up a particular type of elements. We don't necessarily have to assume that it's carbon or nitrogen or phosphorus or whatever. And that specific space, like in our physical world, is different from its environment. And so what we want to do with this specific type of detection method is to use secondary ion mass spectrometry, or SIMS, to take a look at what are the elemental disproportionation that's happening in the sample that we're interested in. And so if you see, just by looking at something like this, you can see that carbon and nitrogen is being sequestered in a way in this blob that could potentially be a cell if we don't know that it's a cell at all. But it points to the fact that maybe there's a process that's happening in this sample that shows that elements are being disproportionately or disproportionated, which points to potential presence of life. So that's one of them. Another one is it's using the fact that the famous quote from some Nobel laureate physicist, the fact that electrons are basically just things trying to look for a place to rest, or life is just electrons that are looking for a place to rest, points to the fact that we can use energy transfer and ways to detect energy transfer as a potential agnostic biosignature. And so this work is being done or is being developed by Harvard University, where these tin oxide electrodes are being used to determine whether or not electrons are being shuttled between two different in a space, and whether or not that particular shuttling is different from abiotic processes. And so in this cyclic voltammetry data or plot that I show here, you can see that the abiotic control has a different shape than the biotic control, the green here, which shows that maybe there's something interesting here going on where life or something similar to life is shuttling those electrons in a more lifelike way than not. So yeah, those are the four main themes that we're focusing on in this work.
And then if so, my follow-up question would be actually, but prior to my question, I want to say, I really like mass spectrometry. I spent my PhD on mass spectrometry, so the following comments has no offense to mass spectrometry.
Okay.
I just want to ask, like, so for example, the few universal biosignature you talk about, complexity, sequencing, the surface features, either above the mineral or cell, or these elemental mapping, and then voltammetry, stuff like that. To me, I feel the sequencing can be done much better if you are talking about DNA, RNA, the sequencer that you are talking about today. And then they're very small. You just need to adapt them to space. Today, even in the commercial mass spectrometry, when people are trying to sequence biopolymers, they really need to have fragments, controlled fragments, in order to do polymer protein sequencing. And then for cell and mineral, it seems like it would be better to have a microscope instead, right? Because I don't think, even if you have a laser attached to a mass spectrometry, it's hard to assess the morphology of a cell or the complexity of the surface feature. For the what elemental mapping, I wonder if you could use spectroscopy. Wouldn't that be easier than using mass spectrometry to do that? And then now this electrode thing, I don't think mass spectrometry can detect the potential of anything. So I don't know. I guess my main point is that based on all the universal biosignature that you mentioned here, I just could think about all different techniques that can actually be used and more effectively detect those biosignatures. And I don't know what could be the unique role of mass spectrometry be in the future. And what do you think of that?
Yeah. So I wanted to clarify that the low power nucleic acid sequencer and the electrode potential detector do not use mass spec. Outside of the example of complexity, and the one where you have to do the elemental imaging using SIMS. Those are the only two that I'm aware of that is trying to use mass spec. Stay tuned for a paper that I'm writing right now on what we can do to agnostify mass specs data. So yes, you're right. And actually, one of the examples that you brought up is another universal biosignature or agnostic biosignature that have been considered before, and that's motility using microscopy. So motility is kind of like a huge feature of life as we know it. And so maybe it could potentially be for life elsewhere as well, but we don't need to know that it's a cell that we're looking for. But the fact that something is moving that is inconsistent with just random processes or Brownian motion. And so that's one other way too. I'm not really sure how to answer your question. I do know that one of the most important things that we need to look for in the future, or needs to design for in the future, is exactly what you said, having the ability to fragment the compounds that are in the sample that we're we are looking at. And so tandem mass spectrometry is a great way to do this. We know that we have tandem MS on MoMA, hopefully on DRAMS. And I know that work is still being in development for designing more modern mass spectrometers that can do fragmentation. But a lot of times, maybe not a lot of times, but sometimes a lot of fragmentation is also not what we want, because we still want to preserve the molecular ion for that sample, for example. And so EI or electron impact mass spectrometry is very, very canonical in our team. Like we use it all the time, or in our community, we use it all the time in GCMS. But a lot of times, like, we don't preserve that molecular ion signature, which is kind of important to distinguish different types of molecules that are in the sample. Anyway, all of this to say that it would be great to get higher resolution data as well. In fact, like the work that we're developing for autocorrelation is only looking at nominal masses, but we want to start to look at how we can use autocorrelation to look at masses with higher resolution, because, you know, amino acid is not just like 57 for a glycine, it's 57 point something X, Y, and Z. And so definitely looking forward to better ion trap technologies, potentially orbit trap technologies, even ICP-MS, the inductive coupled plasma to look at elemental materials, and even FTICRMS, Fourier transform ion cyclotron resonance mass spectrometry. So yeah, that I hope that answers your question. I'm not sure.
Oh, really? Thank you. Okay. Do we have any questions from the audience? Last minute question. Oh, actually, it's already 1 o'clock. Great. Thank you, Lu. This is awesome. Okay.
Thank you so much for attending. And yeah, thanks for the invite. It's so great to be here and so great to talk to all of you. I'm glad we got like 30 minutes of just chatting questions. Great.